Next we have, where are we here? There we are. Maria Pia Carteret. I hope I said that correctly. She has a master's in molecular and cell biology from the University of Bologna and a PhD in molecular medicine at Vite Salute San Raffaele, University of Milan, uh, in the molecular genetics and of renal disorders unit led by Professor Luca Rampoldi, where she is currently a postdoctoral fellow. Today, Maria Pia will be presenting cutting-edge research that I first became aware of last year at last ASN and was just published last month in the Journal of the American Society of Nephrology. And with that, I'll turn it over to you.
Hello, I hope you can see my presentation. Thank you very much, Sharon, for the kind introduction. Thank you for the invitation to share with you our latest results on the effect of calorie restriction on ADTKD UMOD progression. In the lab, we are interested in the molecular genetics of renal disorders, and one of the focuses is ADTKD caused by mutation in the UMOD gene that was already nicely introduced by the previous speakers. I would just like to mention that all the mutations reported so far are missense, and they all are predicted to impact on protein folding. And evidences in patient kidney biopsies and in different in vitro and in vivo model of the disease demonstrated that mutant uromodulin is retained, so it's blocked, in the endoplasmic reticulum ER of expressing cells. In the lab, we generated a transgenic mouse model recapitulating all the features of the human disease. And several years of work allowed us to reconstruct the natural history of the disease. We observed that mutant uromodulin is here retained already at 1 day postnatal in kidney of mutant mice. Starting from 1 week of age, we observed progressive accumulation of mutant uromodulin. We demonstrated that inflammation is an early event of pathogenesis that already occurs at 1 week of age. And starting at 12 weeks, mutant mice start to develop urinary concentration ability defects and progressive renal injury, leading to mild kidney failure at 24 weeks. Now, in this slide, I try to sum up all the knowledge about the ADTKD UMOD molecular pathogenesis, so all the pathways that are demonstrated to be induced by mutant uromodulin expression. Different studies reported the induction of ER stress and the unfolded protein response. Other defects are regarding the mitochondria or increased cellular death of those cells. But today I would like to focus on this phenotype that is autophagy impairment, meaning the block of the degradative mechanism of the cells that was described in different mouse models of the disease, and that in our model is demonstrated by the accumulation of P62, specifically in cells expressing mutant uromodulin. Now, P62 is a specific protein that mediates autophagy, so it is recognized by the autophagosome and degraded along with the substrate. So, the accumulation of this protein in mutant uromodulin expressing cells is a marker of impaired autophagy in those cells. We also reported that in kidney of mutant mice, phosphorylated level of this protein, that is S6 ribosomal protein, and that is a known downstream target of mTOR, the master regulator of the cell metabolism, is strongly induced, again specifically mutant uromodulin expressing cells, suggesting that this could be upstream to the autophagic impairment observed. Now, Based on all these knowledges and trying to integrate an anti-inflammatory strategy with an autophagy-inducing approach, we decided to test the effect of calorie restriction in the context of ADTKD human. This is a dietary intervention that, by acting of a plethora of molecular mechanisms in the cells, has been demonstrated to be beneficial in different settings, characterized by toxic accumulation of toxic protein and chronic inflammation. So experimentally, we used transgenic mice expressing both the wild type or the mutant uromodulin, and those mice were fed ad libitum or with a 30% reduction in calorie intakes, starting from 8 weeks, that in mutant mice corresponded to an early stage of the disease, characterized by sustained inflammation and fibrosis, but still normal kidney function, until 23 weeks of age. We test the effect of this dietary intervention on mutant uromodulin, and we observe that we observe a strong reduction in mutant uromodulin near retention, as demonstrated by reduced colocalization of the transgenic protein with the R marker calreticulin, that correlates with a strong reduction of mutant uromodulin aggregates, demonstrated by the reduction of this high molecular weight smear that you can see in this Western blot, and that is likely corresponding to these huge intracellular perinuclear aggregates that, as you can see, almost disappear after diet. Next, we study lipocalin-2, that is a well-known marker of distal tubule damage, that, as you can see, is strongly induced and again specifically observed in mutant uromodulin expressing cells, and that after diet is dramatically reduced along with the mutant uromodulin aggregates, suggesting that reducing the accumulation of mutant uromodulin can relieve the stress of those cells. And a reduction in lipocalin-2 levels is also observed when we test urine of mutant mice after the diet. Through histological analysis, we also demonstrated an amelioration in tubular dilation, in inflammation, and also in interstitial fibrosis in kidney of mutant mice upon calorie restriction. Now, to assess the effect of the treatment on disease progression, we decided to compare 23 weeks old mutant mice after 15 weeks of controlled diet with 8 weeks old mutant mice, corresponding to the age of the mice at the beginning of dietary intervention. As you can see, ER retention of mutant uromodulin is already present at 8 weeks. It progressively increases at 23 weeks when mice are ad libitum fed, so suggesting disease progression. But in kidneys of mutant mice after calorie restriction, we observe levels of mutant uromodulin near retention that are even lower to those measured at the beginning of dietary intervention, suggesting that this dietary intervention not only prevents the progressive accumulation of mutant uromodulin, but it also clears protein aggregates that are already present. We also studied the kidney function of our mice, and we observe a progressive rise, so a progressive increase of blood urea nitrogen and diuresis levels in ad libitum fed mutant mice, while levels of both these two parameters remain stable and within the normal range when mice underwent the diet. And we also observed that parameter of kidney damage, so tubular dilation, inflammatory cell infiltration, and interstitial fibrosis, at the end of the calorie restriction, show levels that are significantly reduced compared to the starting time point, suggesting that initiating calorie restriction at the early disease stage could, at least in part, revert the already established phenotype. And stemming from this observation, we wonder if calorie restriction could also be effective at an advanced disease stage. For this, we performed a second experiment, feeding mutant mice with the 30% calorie restriction, this time for 24 weeks, and starting at 24 weeks of age, so the age of the mice at the end of the previous experiment. As you can see at 1 year of age, so at the end of the treatment, kidney of mutant mice show a very spongious aspect and a reduction in the kidney weight. But if we look at kidney of mutant mice after calorie restriction, both the morphology of the kidney and the weight are rescued. Regarding kidney function, again, we test blood urea nitrogen and diuresis levels. We observe a strong increase of both these parameters indicative of disease progression. But again, both the parameters were maintained stable after calorie restriction, suggesting that also at advanced disease stage, the dietary intervention can block disease progression. And similar results were obtained when testing again tubular dilation, inflammatory cell infiltration, and interstitial fibrosis. All these parameters progressively increase with disease progression, but are maintained stable by calorie restriction. And we also decided to test senescence-associated beta-galactosidase, that is a marker that was recently reported as a predictor of renal outcome in patients with chronic kidney disease of different origin. As you can see, this marker is already increased in kidney of mutant mice at 24 weeks. It increases and it is maintained stable by calorie restriction, suggesting that this treatment can prevent the worsening of renal damage. Now, we tried to have a more comprehensive view of the effect of calorie restriction, both at early and late disease stages. So we integrated all the parameters tested, and we performed this analysis that is called unsupervised K-mean clustering analysis. As you can see, we observed that all the wild-type mice form a single cluster that is clearly separated from the cluster formed by 8 weeks old ad libitum mutant mice, that again is very well separated from the 23 weeks old 23 weeks old ad libitum mice, underlining this disease progression. But if you look, the 23 weeks old calorie restricted mutant mice form a third cluster, well separated from the previous two, that is closer to the wild type, resembling the effect that we see experimentally on the reversal of the already established kidney damage. For the second experiment, we again observed a single cluster formed from the wild type mice, and the formation of two distinct clusters by the 48 weeks old ad libitum and the 24 weeks ad libitum mutant mice, while the 48 weeks old calorie restricted mutant mice clustered together with their 24 weeks old counterpart, so underlining their very close similarity and confirming the effect of calorie restriction in blocking disease progression. Going a bit more in the molecular mechanism, we also demonstrated that the effect that we observed on the protein, so on the mutant uromodulin protein, is not reflected by any effect on the transcript level, nor by increased secretion of mutant uromodulin in the urine, suggesting degradation as a leading mechanism. So we studied autophagy in kidneys of our calorie-restricted mice, and we observed that the autophagy the marker of impaired autophagy that I showed you before, so P62, is strongly reduced in kidney of mutant mice after calorie restriction. And we also observed a strong reduction of phosphorylated S6 ribosomal protein, so the mTOR downstream target, suggesting that calorie restriction quenches mTOR hyperactivity in mutant uromodulin expressing cells, hence restoring the autophagy impairment in those cells. And additional molecular insight derives from the study of the arphagy, that is the selective autophagy of the endoplasmic reticulum, so the place in the cells where mutant uromodulin is retained, and that is mediated by specific proteins or arphagy receptors. And we observe that genes encoding for some of those receptors are significantly downregulated in kidney of mutant mice compared to the wild type, and they are restored to the basal level after calorie restriction. And among these proteins, I would like to mention just a specific isoform of FAM134B that is encoded by retreg1, and that was previously demonstrated to be transcriptionally induced by nutrient stress to mediate starvation-induced earphagy. So overall, this is the model of our work. So we demonstrated that calorie restriction reduces ER retention of mutant uromodulin, and this is likely downstream to the effect of calorie restriction in restoring autophagy and possibly earphagy in mutant uromodulin expressing cells, with a concomitant degradation of mutant uromodulin. This can, in some way, relieve the overload on the ER machinery, relieving cell stress, and this effect, together with the direct anti-inflammatory effect of calorie restriction, quench fibrosis and prevent the induction of quench inflammation, and prevent the induction of inflammation and block kidney function decline. I would like to finish by thanking all the members of the Molecular Genetics of Renal Disorders unit, all our internal and external collaborators of this project, our founding agency, and all of you for your attention. Thank you.
Thank you so much, Maria Barra. That was just fascinating. I'm really excited to hear questions that folks might have about this potential treatment. I know this is in the early stages of research, but yeah. Lisa, do we have any questions?
Hey, yes, good morning. We do have questions. First question is, per calorie restriction discussion, does the research support or suggest a sort of fasting or periodic calorie reduction to slow disease progression? Surely consuming 70% of daily caloric need would be unsustainable long term.
This is, I think, the greatest limitation of this work. I also think that it's not easy to compare the study that we perform on mice with the direct translation in human, because we cannot directly compare the mouse metabolism with the human metabolism. So for sure, further study will be needed to dissect also the molecular mechanism that we induce with calorie restriction. Also, thinking to, I don't know, employing different strategy such as mimetics of the calorie restriction, so pharmacological compound that can activate the same pathways that we activate with the calorie restriction in the mice. But for sure, further study will be needed. This is really the first step of a very long path, I think.
Well, there's another question. It says, does the calorie restriction research translate to MUC1 variation?
This is a very interesting one. So, in principle, it could work also in the MUC1. Of course, we don't have data by now, but we will be really interesting to test it also in the MUC1 model. In principle, it could be effective because MUC1 should be degraded through the same degradative pathway as uromodulin, at least at the end. So maybe we can act also on the accumulation of mucin 1 by similar strategies. will be very interesting to test.
Another question. What happens if you stimulate I'm probably not going to say this right, autography with urolitine? Urolitine.
Sorry, can you repeat the?
So it says, what happens if you stimulate autography with urolitine? Urolitine.
Yes, I get it. I don't know the the answer. We don't never think to this molecule, but I think that potentially, and actually there are data in cellular model showing that also the pharmacological stimulation of the autophagy acting on mTOR, so the master regulator that I was mentioning, could be efficacy in inducing autophagy and so degrading the protein. So I think that also pharmacological stimulation of autophagy could be effective.
Wonderful, thank you. Next question, would a GLP-1 drug be helpful to facilitate calorie restriction?
This is another interesting one. We are discussing in the lab actually in this class of compound as a part of the calorie restriction mimetics that I was mentioning. It's not clear actually if GLP-1 activates autophagy. There are still scientific debate on this. Potentially it is interesting also because they are now fashion drugs. But I don't know. It needs to be tested firstly on the mouse model to see if we converge on the same molecular mechanism and we can have the same result. We can mimic the same result as the diet.
Well, thank you. Has the possibility that certain foods need to be restricted in lieu of total calorie reduction been explored?
This is another mm, another path that can be explored. We decided to test this type of protocol, so we horizontally restrict the calories. We don't know if there is a specific macronutrient that is contributing to the main effect that we see. For sure, the effect that we see with calorie restriction is really pleiotropic, so we are inducing different pathway. So probably restricting just one specific kind of food, its efficacy in activate one pathway, but not, for example, to have the anti-inflammatory effect of calorie restriction. So these are all experiments that we are really interested to do. Also to dissect the diet, so restricting one by one all the macronutrients, so proteins, carbohydrates, or lipids, to see if we have a specific macronutrient driving the main effects.
Well, that's all the time we have for questions today. And I am just grateful for your being here. It sounds like you've got a lot on your plate in terms of future research, a lot more to learn about this.
Thank you.
So thank you for joining us today.
Thank you very much for the invitation.